record it.
This meeting is being recorded.
Ah, wonderful.
Let me just quickly say we're delighted to have you here today, Dr. Kaczorowski, and we look forward to hearing your talk.
Thank you. It's wonderful to be here. All right, so as many of us know, aging is the leading risk factor for many diseases, particularly age-related cognitive decline and Alzheimer's disease dementia. And we know from heritability studies that genetic makeup plays an important role in determining how an individual ages, whether it's this successful trajectory with little cognitive decline, this more normal aging trajectory with some cognitive decline, or this, a more severe descent into dementia, which is when cognitive impairment becomes so severe it interferes with daily living abilities. And so we're really interested in identifying the genes and pathways that are involved in regulating an individual's trajectory in order to enable a better understanding of the causes of age-related cognitive decline and AD dementia, but also identify potential therapeutic targets. And so here I'm showing you a conceptual framework of how I think and others that individual differences in cognitive decline might emerge. And don't worry, I'm going to walk you through it. So I want to start with this example of normal non-pathological cognitive aging. And in this example, there are molecular, cellular, and global brain changes that correspond to those individual differences in cognitive decline, albeit they're occurring in the absence of classic brain neuropathologies that are used to definitively diagnose Alzheimer's disease. And so these would be amyloid plaques and neurofibrillary tangles. So it's known that these individual differences in normal non-pathological cognitive decline arise by virtue of both genetics and environmental exposures that occur across the lifespan, with things like early life cognitive enrichment, physical exercise, and a healthy diet being considered protective. Some of this is covered in more detail in our recent publications on environmental factors, and I'm not going to have time to discuss this much today, but I'm happy to touch base on it offline. What I do want to talk about is the genetic component, and I'm going to briefly highlight our work with Alyssa Chesler's lab, looking for specific genes that control individual differences in normal non-pathological cognitive decline using the diversity outbred mice as our model system. So just to remind you, the DO mice are a population of outbred mice that are segregating approximately 50 million polymorphisms. And so they're a model of the genetic diversity of humans. And here we use them to identify genetic variants that are associated with working memory decline with ultra-high precision genetic mapping. And so the way that we did that is first, Laura Anderson in the Chesler lab measured working memory ability in a large cohort of DO mice using a cross-sectional design as a part of the shock center phenotyping core. So next, Andrew Willett and Naran Haddad in my lab analyzed these data and found that working memory does decline with age across the DO population. And then they performed genetic mapping and included age as an interactive covariate. And they discovered that there were gene variants in and around the synaptic protein DLGAP2 that was associated with individual differences in this working memory decline. So we next wanted to examine the possible mechanism whereby DLGAP2 might be promoting cognitive longevity in this population. And since DLGAP2 is expressed in the dendritic spines of hippocampal neurons, and changes in spine number and morphology have been implicated in memory and altered in aging, and also because DLGAP2 knockout mice exhibit spine loss in the hippocampus, we quantified spine number and type in CA1 neurons from a subset of the DO mice that we randomly sampled from that 18-month-old cohort following their working memory test. And so what we found was there was a significant increase in the number of thin spines and a reduction in the number of stubby spines in the hippocampus that was indicative of aged resilient mice. Now, notably, these dendritic spine profiles paralleled those reported in human AD resilient cases by the Hershkowitz lab years prior. They just didn't know what genes were involved. So given the relationship between this normal non-pathological aging and signatures of resilience in AD, we wanted to work with Tim Homan and Nick Seafried and ask if our discovery of DLGAP2 in mouse is actually associated with any genetics or omics data from postmortem tissue. And we found that there was a significant association between DLGAP2 at the genetic, at the RNA, and at the protein level. And basically what we found, and you can read it more about it here, is that more DLGAP2 is better. So I would call that a resilience factor. So I think the really exciting thing about this work is that our studies of normal non-pathological cognitive aging in mice do have the potential for nominating new human relevant AD targets. And they provide a system now to conduct rigorous causal tests through genome editing, as well as to screen interventions in a way that just can't be done in humans. So we've also identified several conserved modifiers of normal non-pathological cognitive decline using genetically diverse mouse populations. And we've determined that they play a key role in setting the intrinsic excitability of neurons in the hippocampus, as well as modulate synaptic plasticity, which are two leading candidate mechanisms for information storage in the hippocampus that I'll come back to in a little bit. Work from my lab also developed the first conceptual framework for identifying mouse models of cognitive reserve and cognitive resilience in normal non-pathological cognitive decline, specifically leveraging recombinant inbred mouse populations that I'm going to talk about later, that allow us to resample the same genotype across the lifespan to generate pseudo-longitudinal measures of cognition. And then lastly, collaborative work from the Churchill lab and the DO mouse population led to our important observation that diet interventions that promote lifespan either have no impact or have damaging effects on cognition in these aged DO mice. And we identified genetic variants in the SLC16A7 gene, which encodes a transporter for metabolites in neurons that's associated with individual differences in cognitive performance across this panel. And so, although I don't have time to talk about all of this work in normal aging today, it is really critical because our work in genetically diverse mouse models of normal non-pathological cognitive decline are what served as the foundation for our development of the first genetically diverse Alzheimer's disease mouse population that would allow us to understand how and to what extent human familial Alzheimer's disease mutations interact with genetic background and confer individual differences in cognitive decline. I'm going to bring you back to our model that we talked about previously. And I'm just going to point really quickly to some of our work on resistance to Alzheimer's disease. But first I'm going to there we go. Okay, resistance to Alzheimer's disease. So we define resistance to Alzheimer's disease as the brain's ability to avoid classic neuropathological changes in the brain despite risk factors like inheritance of familial Alzheimer's disease mutations or high polygenic risk. And I'll just tell you that our work in mice and recent work by Patricia Boyle in humans suggests that amyloid burden is not well correlated with cognitive decline. So I was excited to hear about the goals of the ADRC and not being so amyloid-centric, and that it really explains only a small fraction of the variance in cognitive outcomes in the mouse and the human population. And we think this might be one reason why anti-amyloid interventions have not been universally accepted as being successful in clinical trials. So with regards to examining resilience, brain resilience to Alzheimer's disease, this is something that we would define as the brain's ability to cope with those accumulating neuropathological changes in the brain by preventing neurodegeneration. And so Bree Gurdon in my lab has applied an atlas-based quantification approach of labeled neurons and glia across the mouse brain. And to date, we've found no correlation between neuron load and cognitive outcomes in 61 AD mice from genetically diverse backgrounds that she's analyzed so far. She is in the process of registering and segmenting 350 mouse brains. And so I'm excited to update you on that in the coming year. But I will say, because there was not strong evidence for resistance and brain resilience to Alzheimer's disease, for the rest of my talk, I'm really going to focus on what we've learned about the nature of cognitive resilience to Alzheimer's disease. And I just want to point everyone to this recent collaborative publication with Tim Homan and the AD Resilience Working Group that's showing that the genetic architecture of cognitive resilience to Alzheimer's disease is distinct from clinical AD risk. And what that means is that there are, in fact, resilience genes and that they're ripe for discovery. So what is cognitive resilience? So we define individuals exhibiting cognitive resilience to Alzheimer's disease as those that evade cognitive decline that's predicted based on their inheritance of deleterious autosomal dominant mutations that cause Alzheimer's disease and high levels of brain pathology. So in cases of familial Alzheimer's disease that we know result from mutations in amyloid precursor protein or presenilin-1 genes, a delay in the predicted age at onset derived based on your family history of carriers and non-carriers can be used. So if your mother's age at symptom onset was 45 and her mother's age at onset was 43, and you're cognitively normal, but carrying the AD mutations at 60, you would absolutely be considered a resilient individual. In the broader population where we're not talking about autosomal dominant mutations, we actually end up using residuals of predicted cognitive outcome based on biomarkers like amyloid PET scans in order to quantify the resiliency of any given individual. And because resilience to Alzheimer's disease is really difficult to study in human populations, when I started my lab in 2012, we were studying the impact of human familial Alzheimer's disease mutations on cognition in genetically identical mice. And so this is the way most people work, do animal model work related to Alzheimer's disease. And we thought we'd be able to identify molecular and cellular correlates of resiliency. And while we were able to identify molecular and cellular correlates of resiliency in this single strain, utilizing only a single strain is similar to studying Alzheimer's disease in a single human. So the insights gained are unlikely to generalize to other mouse strains, let alone humans. These studies are also not addressing the role of genetics, which we know from human studies explains about 50 to 80% of the variation in cognitive decline. So we developed the first Alzheimer's disease mouse genetic reference panel to better model the heterogeneity of human Alzheimer's disease and to identify the genetic mechanisms that are underlying these individual differences in susceptibility and resilience to AD cognitive decline. And so my lab started with some really high level questions. So the first was, does the introduction of human Alzheimer's disease mutations into genetically diverse mice yield new insights into human relevant resilience factors and their therapeutic targets? And I'm going to show you two examples where naturally occurring mouse genetic variation can be used to identify translationally relevant memory tasks in mice, and also how mapping studies revealed a novel APOE variant that's associated with cognitive resilience. So here's the model that we generated in order to identify modifiers of human familial Alzheimer's disease mutations on cognition. So we bred female 5X-FAD transgenic mice that have 3 mutations in amyloid precursor protein and 2 in presenilin-1 with males from 28 different BXD strains. So these BXDs are a large family of diverse inbred strains that were derived from an initial crossing between 2 common inbred strains, the C57 Black 6J strain, which I'll refer to as the B6, and the DBA2J strain, which I'll refer to as the D2. They're segregating 5 million polymorphisms across the genome. And what you get is a progeny that are a set of F1 AD-BXD strains. So each one contains the same high-risk AD mutations, but whose genetic background is different. This allows us for the first time to understand the contribution of individual genetic variation on Alzheimer's disease susceptibility and resilience in the mouse. We also generated littermate non-transgenic controls that provide insight into the processes of normal cognitive decline. And since each parent strain is inbred, these F1 mice are fully reproducible, and they can be studied across time, in different laboratories, and in response to different environmental exposures, or even therapeutic preclinical studies. Now, to characterize these mice, we measured body weight and working memory longitudinally. And then at either 6 or 14 months of age, these mice go through a detailed phenotyping battery that includes sensory motor tests, anxiety on the elevated plus maze, as well as short-term, long-term memory on contextual fear memory task. After the battery is completed, we are collecting tissue and biobanking it from both the brain and periphery. So people interested in collaborating with those tissues, I'd love to talk more after this. So to determine the age at symptom onset in our mice, we chose to measure working memory longitudinally using the Y maze, because this task depends on the integrity of the hippocampus, which is known as one of the earliest brain regions affected by Alzheimer's disease, and there's little carryover in this task. So the average age at onset of working memory deficits is defined by the chance performance for each strain, and it's calculated and plotted here. So as you can see, the age at onset of working memory deficits varies quite a bit from strain to strain, with some pretty early onset strains, some later onset strains, and then some strains that maintained working memory function for as long as we evaluated them. Again, these would be our resilient individuals. Notably, this variation and the age at onset of our AD-BXD panel looks a lot like variation that we see in human patients who are carrying familial Alzheimer's disease mutations in APP and presenilin-1. So the next thing we wanted to ask is whether or not our AD-BXD population is a good model of individual differences in sporadic late onset Alzheimer's disease, as the genetics are far more complex. So we took advantage of recent studies showing that an individual's genotype across GWAS loci provides an improved estimation of their risk for developing Alzheimer's disease, and this is beyond APOE. And then we were the first to extend this idea to mice. So we calculated a genetic risk score for our mouse strains based on the 21 human AD risk loci that had been identified and have variants across our AD-BXD mouse population. And so to do this, we stratified strains into impaired and unimpaired groups based on their 6-month-old cognitive function. And then we identified the risk allele for each of those 21 genes. The minor allele frequency in this population is 50%. Therefore, we were able to determine the odds ratio at each risk allele in the context of how far it pushed a strain towards cognitive impairment. And from this, we actually calculated a genetic risk score for each individual strain. And we found that the genetic risk score was significantly associated with late disease cognitive performance that we measured at 14 months using contextual fear acquisition, but it did not predict age at onset or disease phenotypes in our non-transgenic BXD littermate controls. And so I think these results are really important because they suggest that contextual fear acquisition is a task that's sensitive to variation in human relevant AD risk genes, and therefore it has the potential to be used as a translationally relevant cognitive assay for preclinical studies, which historically has been a major problem in the context of AD preclinical work and translating to humans. So then we performed genetic mapping of this trait, and we identified a single significant QTL on chromosome 7 that explained 21% of the phenotypic variance. The D2 allele was defined as the risk genotype, and our QTL interval was syntenic to the APOE locus in humans. And so given the known coding variant in the receptor binding domain of APOE shown here, we stratified our strains based on their APOE genotype and plotted cognitive performance. And we found that cognitive performance tracked with APOE genotype, where heterozygous D2 carriers performed more poorly as compared to the homozygous B6 carriers. Now, because the mouse APOE variant was not predicted to be damaging based on an amino acid change from glutamic acid to aspartic acid, we wanted to examine the consequences of this variant in the context of APOE protein as a whole. So in collaboration with Zach Levine at Yale, molecular dynamic simulations were run on the B6 and the D2 APOE isoforms in both water and in the presence of lipids. And what they found is that the lipidated APOE isoforms from B6 and D2 mice inhabit different states. And when you compare the portion of lipidated D2 APOE proteins remains somewhat closed, like the APOE4 isoforms that elevate Alzheimer's disease risk. The other thing that was really interesting is that the B6 state, so our resilient APOE isoform in the mouse, was unique, and it didn't look like E2, E3, or E4 in the humans, and I'm going to circle back to this in a moment. So to directly test if that APOE D2 variant was conferring susceptibility to cognitive decline, we worked with Rick Mazer to use CRISPR genetic engineering and develop a B6 mouse with that D2 APOE allele. And then we crossed this to the 5X FAD mouse in order to generate 4 groups. So non-transgenic B6 and non-transgenic B6 with the D2 APOE, with and without that 5X FAD Alzheimer's disease transgene. And what we found is that those 5X FAD mice with a single copy of the APOE D2 allele were more susceptible to cognitive decline compared to the 5X FAD mice on a B6 background. Of note, this genetic mapping suggested that 2 copies of the B6 allele confers some resilience to familial Alzheimer's disease mutations. And we actually did this work in 2017. And so what I think is really interesting is this region of APOE was later rediscovered in a woman who was a carrier for presenilin familial Alzheimer's disease mutations, and also homozygous for the APOE Christchurch mutation that is considered to be protective. And so Zach and I are now running the molecular dynamic simulations to determine if mouse APOE B6 isoform is similar to the human APOE isoforms containing that Christchurch mutation. And we're hoping to have those results soon. Taken together, this work really demonstrates that we can identify and validate human relevant modifiers of familial Alzheimer's disease that are rare, even before they're discovered in the humans, which is really cool. But given that there are a variety of mechanisms whereby the APOE genotype could confer resilience to Alzheimer's disease, we wanted to take an unbiased approach and try to identify the cell types and gene expression profiles in the hippocampus of these mice using single nuclear RNA sequencing data. And so here I'm showing you that the vast majority of differentially expressed genes from those 5X FAD mice with and without that single copy of the APOE D2 allele are actually in excitatory neurons of the hippocampus, and they're mostly in the dentate gyrus and the CA1 region. And our pathway analysis suggested that neurogenesis, synapse organization, metabolic function, and neurotransmitter transport were altered in the 5X FAD knock-in mice. And so the reason I think this finding is so important is that it's the first demonstration of a role for APOE in promoting resilience to familial Alzheimer's disease mutations through their effects in excitatory neurons. And this differs from the published roles for APOE in glial cells. And I think that that's really an avenue that we're going to continue to pursue in my lab. So I'm going to quickly summarize this first part of my talk by telling you that I think genetic background is incredibly important. It makes for more translational models of human disease, and it also provides the tools that we need to identify and validate resilience factors and their mechanisms of action. And I think this is really going to open up a brand new set of interventional strategies, which I refer to as resilience-based therapeutics. And so next we wanted to ask, outside of APOE, where does risk and resilience to Alzheimer's disease originate in the mouse, at the molecular, at the cellular, and at the anatomical levels? And I'll just quickly tell you before I'm going to show you that our data point to specific hippocampal and cortical neuron types that seem to remodel in response to microglial activation and proliferation. So first, Sarah Hewer, when she was a graduate student in my lab, was really keen to use all that behavioral data I talked to you about in order to generate decline scores for each AD-BXD strains, and then integrate that cognitive data with gene expression and genetic data across the AD-BXD population to nominate pathways and cell types that best explain individual differences in cognitive outcomes in mice, and then cross-check them against the human data. And so what she did was take RNA sequencing data from the hippocampus, this is bulk sequencing data, and she built co-expression modules using the consensus clustering algorithm SpeakEasy. And she identified 31 discrete transcriptional modules, which are shown here. The heat map is showing you how the modules relate to each other based on their overall expression, gene expression. And then the second heat map in gray is showing you the enriched cell types from our deconvolution analysis using single cell RNA sequencing data. And then she also performed go-enrichment on the genes within each one of those modules to get an idea of the biological processes and pathways that were represented. And together, these modules covered transcriptional networks that were enriched for many disease-associated cell types and biological functions. So next, she wanted to know, how do they relate to individual differences in cognitive outcomes? And so what she found was that 16 of the 31 modules that were significantly correlated with at least one cognitive measure after correction for multiple comparisons. And you can see that here. She looked at working memory, short-term memory, and long-term memory. Next, in order to identify the modules that were explaining the most variance in individual differences in these outcomes, she performed a linear regression. And as you can see, there are a number of gene modules whose differences in expression explain a fair amount of variance in each cognitive measure. So those in red are increased in glia and mostly microglia in susceptible strains, where those in blue are increased in neurons from resilient strains. And so these results told us that in addition to microglia that a lot of people are studying now in the context of Alzheimer's disease, we still need to focus on neurons in order to identify resilience factors. That said, we didn't really know which hippocampal neurons we should specifically be focusing on. And so given that the hippocampus is comprised of a large number of neuron classes and subtypes, we thought to use single nuclear sequencing data to try and link co-expressed resilience genes to their cell type of origin. And to identify cell type specific transcriptional signatures that were associated with cognitive resilience, we performed single nuclear RNA sequencing on hippocampal samples that were collected from 12 AD-BXD strains and their non-transgenic counterparts, where 6 were defined as susceptible and 6 were defined as resilient relative to their population mean. And then the nuclei were isolated using manual dissociation and loaded for capture using the V3 chemistry and ultimately sequenced. The gene expression profiles were analyzed from 220,000 nuclei across all of these samples, and then they were clustered, and I'm visualizing them here. The cell type identification for each individual cluster was then based on looking for the overrepresentation of cluster markers using the Hippo-seq data. And what I'm showing you here is that all of the major cell types in the hippocampus were represented across our 32 unique clusters. We then subclustered the excitatory neuron class, which revealed 21 cell type clusters that reflected neuronal diversity expected in the hippocampus. And so one of the first observations that we made by comparing differentially expressed genes from susceptible AD carriers to their non-transgenic counterparts is robust changes in the gene expression of the dorsal dentate gyrus and CA1 neurons of the hippocampus. And these were, for the most part, absent in resilient strains at both 6 and 14 months of age. And so this led us to speculate that Alzheimer's disease-induced gene expression changes underlying cognitive decline were somehow being buffered by the genetic backgrounds that actually conferred resilience to the AD mutations. When we specifically focused on differentially expressed genes in excitatory neurons of susceptible versus resilient AD mutation carriers, we see that the top pathway is ribosomal protein synthesis, which is really interesting to us because the candidate mechanisms of information storage that I spoke to you about earlier, being intrinsic neuronal excitability and synaptic plasticity, these are known to be protein synthesis dependent. And this actually provided a really strong premise for our new R01 that's aimed at using MetaRS and TurboID mouse models to identify newly synthesized proteins in a cell type specific fashion in AD resilient mice with the goal of identifying novel resilience-based targets. Lastly, we integrated the transcriptional profiles of glial cells, which we know are involved in neuroinflammatory responses to amyloid, with our neurons into NicheNet. So NicheNet is a method that allows us to predict ligands from astrocytes, neurons, and microglia that are acting at their receptor or signaling targets in those two resilient cell types I told you about, the dentate gyrus and the CA1 neurons. And so this method basically combines cell expression data with prior knowledge on ligand receptor pairs, as well as signaling and gene regulatory networks. And so what I'm showing you here is just the top ligands that we're predicting will promote cognitive resilience to Alzheimer's disease mutations through their influence on dentate gyrus and CA1 neurons. And I wish I had more time to discuss these here because I think the cell-cell interaction is going to be critical for conferring resilience to Alzheimer's disease. But suffice it to say, our results provided a pretty short list of potential secreted ligands that we want to directly test on their downstream targets. And so to do this, we established patch sequencing in the lab with help from the O'Connell group. And so patch sequencing is a modification of whole cell patch clamp protocols that enables mRNA sequencing of the cell contents after electrophysiological recordings from individual neurons, as well as capturing their morphology. And this is going to allow us to back apply some of those ligands of interest that I just showed you on the last slide in order to evaluate their effects on neuronal function and structure, as well as their consequences on gene expression in a cell type specific fashion. And so this is just proof of concept data showing the functional readout of 8 neurons from the hundreds that we've recorded so far, and the expression of tens of thousands of transcripts and nearly 8,000 genes that we can sequence from a single nucleated patch that we pulled from a neuron using smart sequencing. And so to just summarize this part, I think that the insights that we have gained are really important in terms of pointing us towards specific neuron types and pathways that are associated with cognitive resilience. And I think given that they are likely influenced by glial cells from the work of Sarah Hewer and others, we're now positioned to test these using state-of-the-art patch sequencing and other techniques. Okay, so next we wanted to determine the extent to which these resilience signatures are conserved in humans and prioritize candidates that have a likely therapeutic value. And so to do this, we generated single nuclear RNA sequencing data from mouse brain tissue that contains the prefrontal cortex. And so we targeted the prefrontal cortex for two reasons. The first is that hippocampus-dependent memories are transferred to this region for long-term storage, which is a process that is disrupted in Alzheimer's disease, and because comparable data had been generated from susceptible and resilient AD human cases from our collaborators from the postmortem tissue. And so in order to examine the alignment between the mouse and the human single nuclear data sets, raw data from the human and mouse samples were integrated and analyzed using the package Liger, which was developed by the McCosker lab. And I'm showing you here that after integration of the data and two-dimensional visualization, you can see that the human and the mouse nuclei overlap in terms of their organization into cell clusters. These clusters comprise all of the major cell types in the prefrontal cortex, the vast majority of which have a cross-species alignment score of 0.65 or greater, and I've bolded those here. And this is indicative of well-conserved cell types. Next, we looked at differentially expressed genes from susceptible and resilient humans and mice, and this pointed us quite clearly towards cluster 20. So cluster 20 had the most upregulated genes in resilient individuals in both the humans and the mice. And using meta-neighbor analysis and self-census network data sets from the Gillis Lab and the Brain Initiative, we were able to identify cluster 20 as layer 4-5 isocortical neurons. And our pathway analysis revealed a likely role for mRNA metabolism, CNS development, neurogenesis comes up again, viral processes, and protein localization, with overlap of several genes and more than one functional pathway. So you can imagine how variants in a particular gene or set of genes can actually influence multiple functional pathways. So among those upregulated resilience genes from cluster 20, a total of 12 of them, 11 from the mouse and 1 from the human, had previously been nominated as AD targets on Agora. So Agora is an interactive web-based tool. It was developed by Sage Bionetworks in collaboration with members of the AMP-AD consortium, which is comprised of academic and industry teams. It includes Model AD, Resilience AD, and Treat AD. And the thing that matters about Agora is that it's hosting extensive druggability information that's developed by industry partners. And I'll say of our Agora nominated targets, only one had a good druggability profile. In addition to those that were previously nominated, we identified a total of 61 new targets, and these were subsequently validated by us using human proteomics data from the Emory team. Of these 61 targets, I'm showing you that only a small fraction of them are predicted to be druggable and safe on Agora. And I think this really drives home the point that we need to continue to search for new gene targets and mechanisms of cognitive resilience if we're actually going to be able to find one that's going to be effective and safe. And so to summarize this third part of my talk, our work is really the first to directly test the alignment of mouse and human prefrontal cortical cells in a cell type specific fashion. And we found that the vast majority of them are well conserved. And I think this is telling us that that genetic and phenotypic variation in our mouse population is what's key to modeling human disease. And that prior criticisms of the poor translational value of classic AD models had far less to do with the species and way more to do with major limitations of studying any disease in a single genetic background. We also nominated layer 4-5 cortical neurons as key players in cognitive resilience to Alzheimer's disease and proposed several new gene targets that may promote resilience with good druggability and safety profiles. And we'll be testing some of those shortly. Okay, so next I want to show you how we're moving from a single gene target to the discovery of small molecules with good selectivity and safety profiles. So as some of you might know, in 2017, we identified the non-canonical cation channel TRPC3 as a potential target to treat Alzheimer's disease. So this channel was overexpressed in brains of impaired, aged, and AD mice. And it was also correlated in postmortem AD tissue to risk. Using viral-based gene therapy tools, we showed that a 20% knockdown of the TRPC3 protein in the hippocampus could prevent cognitive decline in normal aging mice, as well as AD mice, which I'm showing you here. But a selective and safe antagonist was not available. So as an academic lab, we weren't really positioned to run a drug discovery program around this target. But with the help of a number of people at JAX, we were able to partner with medchem experts in pharma, thanks to generous funding from both JAX as well as several angel investors. And so without going into too much detail, this team set up a high throughput screen of over 109,000 compounds. And from that, we prioritized 624. Running dose response curves, there were really 2 series that emerged and were chosen for lead selection and optimization. And from that, we arrived at 3 really interesting molecules, which unfortunately I can't share the structure with you. But what I can share is that these 3 molecules have favorable properties in terms of their binding profiles to TRPC3, their PK/PD in both human and mouse liver microsomes, as well as their selectivity. So the other thing that we got really excited about is that after IV dosing in the mice, these compounds also penetrate the brain well. So with the help of Kat Lutz and in vivo pharmacology, we're going to plan to start testing these compounds in mice soon. Now, the discovery of TRPC3 came out of years of work evaluating cognition in very large cohorts of aging and AD mice in vivo, which is incredibly slow and expensive. And so if anybody knows me, you'll know that I'm pretty impatient when it comes to generating and interpreting data. And so in order to accelerate the unbiased discovery of novel resilience factors, we wanted to develop a 3D co-culture system that exploits established differences in the onset and progression of cognitive symptoms in our AD-BXD panel in order to establish a therapeutic screen. And so with new funding from the Alzheimer's Disease Association and help from the Kaplan Lab at Tufts, Maria and I are now developing a 3D model of cognition to perform an unbiased screen of compounds using more sophisticated readouts that were derived from our single cell sequencing work that include neuronal differentiation, changes in morphology like spine morphology, leveraging patch sequencing to link gene expression to functional changes, as well as excitotoxicity. And so I think that I'm also really keen to look at how the readouts are altered, especially in response to those ligands that were predicted to be secreted by neighboring cells and influence neuronal function, particularly in the dentate gyrus and the CA1 neurons. Thanks to the amazing work by the Reinhold and Pear labs here already at JAX, we have a roadmap to pursue these cellular models of mouse phenotypes. We're just doing it now in the context of Alzheimer's disease. And so last but not least, once we have those targets, how are we going to know what mouse models to test them in? And so in terms of picking AD-BXD strains for our drug testing, we asked how well hippocampus gene expression profiles align with those that were recently published from human AD profiles from Bin Zhang's group. And so what Bin was able to do is take hippocampal transcriptional profiles from a large set of patients and do unbiased clustering in order to identify 3 different major subtypes of Alzheimer's disease. So the first was human subtype A. It stands for atypical, and it's enriched for synaptic genes. The second is human B for a more tau-related phenotype. And then human subtype C is sort of a classical phenotype that has an amyloid immune-like gene expression response. And so what we could do then is use our RNA sequencing data from the hippocampus of 14-month-old AD-BXD strains and apply supervised clustering And what we found was that there was actually high alignment between the molecular profiles of specific mouse strains and those human subtypes from each of those 3 main clusters that I'm showing you here. Remarkably, we find enrichment of female AD-BXD strains that are aligned to the classic amyloid AD human subtype, which parallels the higher incidence of females with AD. And that tells us that sex can actually push a strain into a different AD subtype. So for example, AD-BXD strain 75, its transcriptome in females is telling you that it's closest aligned to the classic AD human subtype C, whereas the male actually models the tau-related subtype B. So given this information, our preclinical studies are going to start with the collection of 3 mouse strains with profiles that are closest to one of each of the human subtypes with both males and females. And then we're also going to leverage this really massive study that was run by Amy Dunn in my lab, where she examined the effects of high-fat diet and genetics on 6,000 mice longitudinally. And I had to summarize her work in only 1 slide. But basically what she found is that both sex and genetics regulate an individual's response to a high-fat diet. So I think, our data has now shown that even this classic risk factor like high-fat diet isn't bad for everyone, and its impact on disease risk really depends on genetic background. And so it highlights how we need to start doing more personalized precision medicine approaches, even in animal models, because what's going to work for individuals of these strains to the right may not work for these individuals on the left, both in terms of lifestyle interventions, but also pharmacological treatments. And so if I put all of these pieces together, this last slide is just meant to illustrate the current and future work that is being pursued in my lab. So on the top left, in addition to aligning the AD-BXD strains to human Alzheimer's disease human subtypes, we're also generating mixed pathology models for AD-RD with Kat Lutz and Kristen O'Connell, as well as Q111BXD models of Huntington's that we're going to maintain on genetically diverse backgrounds. And I think our goal here is really just to define models of susceptibility that we can use in order to test resilience-based interventions and evaluate how well they generalize to new models of late onset Alzheimer's disease, like those from the Model AD Consortium, but also other neurodegenerative diseases like Parkinson's and frontal temporal lobe dementia. And then here we'll continue to take our multi-omics approach in order to identify resilience mechanisms for each one of these subtypes. We're also going to work towards validating polygenetic risk scores in mice that are aligned to GWAS loci for Huntington's disease traits in collaboration with Jim Gisela at Harvard as part of our new R01, as well as psychiatric traits in collaboration with Rob Sweet as part of PsychAD. And so I think we're going to know that mouse assays are translationally relevant when they're sensitive to genetic risk scores that are derived from Huntington's, psychiatric, motor, and sleep-related GWAS hits. And I'm really excited because I think this might help us identify global resilience mechanisms to aging that really cut across neurodegenerative diseases should they exist. And so I'm also, we have new funding, like I told you, to continue to design in vitro 3D models of mouse AD subtypes that will allow us to test some of our novel targets, but also perform drug discovery screens using gene expression and electrophysiology and cytokine and metabolic readouts. And now we're really positioned to test these compounds in relevant AD subtypes with both behavior and MRI readouts, which I didn't have time to show you today, but I'm happy to talk about, that we really need for target engagement in these preclinical studies. And we plan to openly share all of this data on the Knowledge AD portal and AMP-AD website so that it can be evaluated by the greater scientific community. And with that, I'll just thank you all for your attention. I wanted to also thank the JAX Angel investors who've been advising and supporting the TRPC3 drug discovery program. which is actually my first attempt at bridging the gap between our basic science discoveries and therapeutics. So it's been incredibly challenging, but also really rewarding. I also have to thank Tony Ebnen for his generous support, and also for sharing his really deep network of academic and industry contacts that have come in incredibly well, like very handy. I want to thank my entire lab, past and present. These guys are really a lot of fun and they're absolutely brilliant. I have to thank the scientific services at JAX, especially our Center for Behavioral Analysis, which is basically like a mouse hospital, and also the Single Cell Biology and Protein Sciences group. Of course, the leadership at the lab have created the kind of opportunities that every scientist really dreams of, and so I want to thank our leadership. Certainly my external collaborators that make this mouse to human translation possible and really, really fun. And then the faculty at JAX, both my mentors, Gareth and Alyssa, but really all my collaborators at the Jackson Laboratory. And finally, I'm going to thank all of the funding that we have that we've been blessed to receive to do all of this work. And with that, I can take any questions. And I'm very sorry I got off to a late start.
Thank you so much, Dr. Kaczorowski, and that was an absolutely beautiful whirlwind through your work. Thank you for that, and no apologies are needed from you. It's our fault that you started late, and I recognize that we've lost a good chunk of our audience given that we're past the hour of 1, but certainly those of you who are still here, we have time for a few questions. If you would like to ask Dr. Kaczorowski a question.
I can stop sharing too so I can.
Let me lead off, and I know this is only a very small, there's so many things, small bit of what you're doing, but these cognition in a dish using these collagen grids in which you're putting hippocampal cells, presumably they create a network of non-neuronal cells and neuronal cells that, that engage in activities that would, to you, replicate to some way the hippocampal function, or no?
Yeah, I think they will. I mean, I think the advantage over 2D models is that we'll be able to evaluate spine morphology much more easily. And then also the patch sequencing and synaptic throughput for us as electrophysiologists, slice of electrophysiologists is much easier in 3D than 2D. And so I think we're really excited about that. I will say, having seen Jack's recent paper making organoids, right? I think that's also a really successful way to approach making, modeling complex cell-cell interactions that are affected by genetic factors that we can use readouts for drug screening.
Thank you. I think given the late hour, Catherine, and since we have lost literally 80% of our audience, and we had a good turnout, and since you have a busy day ahead of you, including starting at 1:30, I will note there was the HSP90 gene among your list of genes that are in the neurons that might be affected by microglia, and you're meeting with Matthias Trutman very shortly, so he will want to talk to you about that. Let me thank you again for this beautiful tour de force presentation, and thank you for taking the time to tell us all about this.
Yeah, wonderful. Thank you.
All right. Bye-bye.